Hello students, in this video we'll study on cellular totipotency. So if you define the cellular totipotency, it is a potential of plant cell to grow and develop into a whole new multicellular plant is called or is described as cellular totipotency. In other words, the property of a single cell for differentiating into many other cell types is called a totipotency. It is a nature in plant cell but not observed in animal cells. This is a property which is found only in the living plant cells, not in the animal cell. Exception being the stem cells in animals. The term totipotency was coined in 1901 by Morgan during the culture practice. An explant is taken from a differentiated or mature tissue is called primary cells. To show the totipotency, such as mature non-dividing cells undergo changes which revert the completely differentiated cells into a meristematic state. Usually it is called as a callus state in plant tissue culture. This phenomena of reverting back of mature cells to dividing state is called dedifferentiation because already differentiated cells, mature cells, are dividing back to a meristematic stage. We call it as dedifferentiation. Now, these dedifferentiated cells have the ability to form a whole plant or plant organ. This phenomena is referred as redifferentiation. So, picture he can see here the single cell which is divided into a continuously dividing state called meristematic state, and each cell has the potency to develop into the whole plant. Again, this is the picture of where the callus cell, which is from the carrot. From the carrot, a single cell can be obtained from callus culture or from uh, the direct tissue, and these the cells which are inoculated into the nutrient media, it will form a callus structure, undifferentiated mass of cells. From the callus, you can develop into embryo. This is a picture of early embryo and the late embryo, which germinate to form the whole plant. Let's see here. We can see here the single cell taken from the explant. It has the potency to develop into a whole plantlet. So this phenomena or this ability of the single plant cells to develop into whole plant is called as a totipotency. Explant, it is from mature non-dividing or differentiated cells to form a callus undifferentiated and meristematic cell. This step is called dedifferentiation. From the callus, they again has to form a differentiated cells or new plantlets, and this process is called as redifferentiation. Here we can see the excised plant leaf which is formed into callus or a single cell formed into callus where they are induced to form the shoots or induction of shoots and to develop into the whole plantlet. Dedifferentiation and redifferentiation are the two inherent phenomena involved in the cellular totipotency. It is uh, a clear differentiation is that basic event for development of plants, and it is also referred as cytodifferentiation. To express the totipotency in a plant cell, a differentiated cell first undergo the phenomena of dedifferentiation and then undergoes redifferentiation. As I told you, reverting back the matured tissue into meristematic stage is called as dedifferentiation. And then forming again the differentiated tissue is called as redifferentiation. Usually, the dedifferentiation of the explants leads to formation of callus. However, the embryonic explants sometimes result in the differentiation of completely differentiated tissue like roots and shoots without an intermediary callus. Again, the flow chart shows that the process from here to here, that is, from the mature leaf, the leaf tip or a piece of the leaf, it can be developed, that is, the leaf cells, which are isolated with a homogenizer and culture to form a undifferentiated mass of cells, is called as dedifferentiation. From the undifferentiated mass of cells, where they are developed into shoot buds. Formation of shoot buds and shoot buds isolated washed with again subculture to form the whole plantlet, and this step is called as redifferentiation. It is dedifferentiation and this is redifferentiation. The plants have such a quality that even a highly mature and differentiated cells have an ability to revert back to meristematic state. This property of totipotency of plant cells indicates that even undifferentiated cells of callus carry the essential genetic information required for regeneration of a whole plant. All the genes that are responsible for dedifferentiation or redifferentiation are present within the individual cells, and they become active for expression under adequate culture condition. As the totipotency cells are the basis of whole plant tissue culture technique, so by the exploitation of this potential of plant cells, biotechnologists are trying to improve the crop plants for other commercially important plants. The totipotency in different plant parts. The somatic cells in the plant body are totipotent. The living plant cells have the ability to regenerate, and the dead cells. The cells which lack cytoplasm and nucleus, like tracheids, vessel elements, are not totipotent in nature. Different plant parts have the different totipotent abilities. For example, in tobacco plant, the bud formed by the in vitro culture of the epidermis of different regions of the tobacco plant. Another example to add this here is totipotency of crown gall cells. Crown gall means it is a disease caused by the infection of agrobacterium in tumefacious to the dicot plants, which have the capacity to grow as an unorganized mass of cells under normal condition. However, the whole plant can be recovered the from that culture. That is, this is the crown gall disease. From the single cell can be induced to form the formation of whole plant in the in vitro culture. Thus, it is clear that totipotency is not similar in all plant parts. So, where this totipotency, this nature of the plant cells can be applied? It is applied to prove to be a boon to mankind, as it is a basis of plant tissue culture. The plant tissue culture exploits this technique properly, this property for plant cells to attain the commercial benefits. Like it has a potential applications in the crop plant improvement, micropropagation of commercially important plants, production of artificial or synthetic seeds, and it helps in the conservation of germplasm or genetic resource. It has the ability to be utilized for haploid production, applied in producing somatic hybrids and hybrids, helps in the cultivation of those plants which or whose seeds are very minute and difficult to germinate, also helps to study the cytological and histological differentiations for high scale and efficient production of secondary metabolites. And the genotype modification can also be possible by using this nature of totipotency. Okay, this is about the cellular totipotency in the plant cells. Thank you for watching.